Hello everyone, my name is Qingyang from the Department of Statistics and Data Science at UCLA. And today I'm going to talk about a collaborative project with Dongyuan, who is my lab mate, which our project is called scDesign3, which may sound familiar to you because my advisor talked about this about an hour ago. And I will try to minimize the overlapping contents between my talk and her talks. And the first author of this project, Dongyuan, was not able to come here due to the schedule conflict, so I will do the presentation today. And for today's talk, I will be more focused on the motivation and the detailed introduction of the functionality, about the functionality for our simulator. And for the introduction part, as we all know, in the single cell field, we already have a massive data set and various computational tools. On the left figure, we can see that we already have so many cells being sequenced, and these cells can come from various cell states, including discrete cell types, continuous cell trajectories, and also we can have the spatial location of the sequence samples due to the spatial omics technologies. And on the right figure, we can see that based on this scRNA-tools.org, there is already more than 16 hundreds of computational tools being developed to analyze those sequenced cells. So for now, we want to ask if there is a unified probabilistic model for us to interpreting this single cell and spatial data. And if we can have an all-in-one simulator which can provide us the ground truth for comparing the various computational tools that is already being developed. So we summarize some of the challenges in developing those kind of tools and in modeling the single cell and spatial multi-omics data. First of all, is the high-dimensional problem. As we all know that for example, for the single-cell RNA-seq data, there should be around or at least 10,000 of genes, and for other features, this number can be even larger. And these features would have a complex correlation structures that we want to capture in the synthetic data. And for the sequenced cells, they can come from diverse covariates, including the discrete cell type, continuous trajectory, and the spatial locations from the spatial transchromatic data. And the different omics the data come from different omics may follow different distributions when we're modeling them. And lastly, we also want the transparency in our models, so the model parameter can be interpretable and we can find some biological patterns through the model parameters. So here comes our work at scDesign3, which is published this year on Nature Biotechnology in May. And I will just go more detail about this plot, which you may see this before. And the first part of the functionality is the simulation part, which we can simulate the cells from diverse cell types, which are discrete or continuous trajectories, and also across different spatial locations. And our simulator can also simulate data from different modalities, including the scRNA-seq data, TAC-seq data, site-seq, and DNA methylation data. And our model can incorporate in the consideration of the different experimental design. For example, you can have patients with different gender, age, and different health conditions. And also the cells can be sequenced from different batches, and we will be able to capture, or you can remove this batch effect during the data simulation process. And the next part is the interpretation part, because we are a probabilistic model. So we have the model parameters, which will be able to reveal for example, the difference in gene expressions across different cell types, along the pseudotime trajectories, and the gene expression patterns across different spatial locations. And also, we provide the unsupervised matrix AIC and BIC to reveal the goodness of fit of the covariate to the model. So in here, you can see the first, so this one is we'll want to illustrate an example of the bad pseudotime, because you can the pseudotime is all mixed up, which will have a larger BIC compared with this pseudotime, which shows the clear patterns of changing colors. And also similarly to this cell cluster labels, in here the yellow one and the purple one are mixed together, but for this good one, they are well separated. So this one will have a lower BIC. And last one, since we have some model parameters, you can also modify the parameters based on your needs. For example, we can specify some predefined differentially expressed genes across different cell types. So you can manually enlarge the difference between the gene, mean gene expressions between different cell types, or you can make some genes be non-D genes, which means their mean gene expression across different cell types are identical. And lastly, you can generate the synthetic null data, which are cells from one homogeneous population based on the reference data set, which could be cells from the heterogeneous populations. And for the next part, I will just show some results since Jessica already discussed the details of the method, so I won't repeat the method again. I will just show some results that she didn't show. And the first one is our comparison with other existing simulators. Some are deep learning based, some are not. And in here, we first divide the data set into the training data and testing data. And for the training data, we inputted this training data into 5 different simulators, including scDesign3 as the reference data set. As you can see here, our simulator scDesign3 will capture these continuous trajectories, while some other simulators make this data more discrete-like instead of showing the continuous trajectories. And that's the visual check of the performance. And then we also have the quantitative values on this MLISI, which is the local inverse Simpson's index, which reflect how well this simulated data mixed with this real data here, which takes value between 1 and 2. The closer the value to 2 means a better performance. So from the quantitative perspective, our simulator also has a better performance compared with the other simulator. And this is a single trajectory case. And this one will be a bifurcating trajectory case. So in here, the testing, the real data shows a bifurcation in the pseudo time, and our simulator was able to capture this bifurcation, but while the other simulator was not able to clearly capture this bifurcation patterns, and some may look like a single trajectory instead of the bifurcating trajectories. And the next one is our simulated data for the spatial transcriptomic data. Here is 3 genes, and the color is their expression levels, and the dots is the cells at different spatial locations. So in here, there are 3 more spatially, 3 genes that has more clear spatial patterns, and our simulator was able to capture these spatial patterns of gene expression across different spatial locations. And this one is a simulated data set given a site-seq data, which we were able to capture the RNA and protein abundance in the blood cells. And this one is more about the interpretation part of interpretation functionality of scDesign3. In here, we first use a synthetic data set, which can provide us a true time of the cells. And then we also have some inferred pseudotimes. And in these 3 different colored dots are 3 sets of pseudotime inferred by different pseudotime inference methods. And there is 10 black dot here, which is the pseudotime with various degrees of perturbation. So for example, this one is completely the true time, and this black dot is we have 10% of the pseudotime being shuffled, meaning that the quality of this set of pseudotime will be worse than this set of pseudotime. And for the next 8 points, there are 20%, 30%, and up to 100% pseudotime being shuffled, so which reflect the quality is become worse and worse. And we reveal these quality changes using the R-square between the true time, which we obtain from the simulated data set, and the inferred pseudotime, either from the pseudotime inference method or the manually shuffled pseudotime. And here we can see our unsupervised matrix BIC agrees with this supervised R-square, which can illustrate the quality of the pseudotime being inputted to our simulator. And here is how we can generate the synthetic node data using scDesign3. As you can see, for the reference data sets, there are cells from two different cluster cell types. And then one way we can simulate is to keep this difference between the cell type, which could be an alternative hypothesis that the cells actually do come from two distinct cell types. And then we can also simulate this data under the null hypothesis that the cells are from one homogeneous population instead of the two cell types. In here, the synthetic data, we are filling the gaps between the two distinct cell type from the reference data. So they look from the same homogeneous populations. And lastly, recall that for scDesign3, we first fit the marginal distribution independently for each feature, and then we use a copular technique to join those independent marginal distribution together to a joint distribution. So here we use a copular technique which can reveal the biological difference between the cell types. So if you are using the Gaussian copula, you get a different gene correlation matrix for different cell types. And then if you are using the Wang copula, which utilize the Wang structure, try to connect different genes or features through the bivariate copula. So they give you a structure about how the genes are connected in these fine structures. So in here, we can see the different cell type have a different Wang structures, which may reveal the biological differences between the cell types. And lastly, I would thanks to the first author who couldn't come here, which is Dongyuan, and our advisor, Dr. Jing Jessica Lee. Here is a link to our paper and our software. The scDesign3 was initially implemented in R, but now we also have a Python implementation, which is called scDesign3 PY. And please feel free to contact us if you have any questions, comments, or suggestions. Thank you.
Questions?
Yeah.
It's a quick question, but are you able to generate sparse data? Because we have because many of the simulation and Mulid ODE, for example, generates very dense data, but maybe feature of that.
You mean sparse data?
Yeah, is the data sparse enough like what we observe in the real single cell?
Yeah. So the question is our simulator able to generate sparse data sets?
Like, is it sparse enough, similar to the sparsity of?
Yeah, we check various characteristics of the synthetic data set, which can be found in our manuscript, and we found our simulator will mimic all the characteristics from the reference for the reference data set.